Hello everybody, my name is Omar Zadan and today we're going to be explaining spermatogenesis, a topic important for the BAU entrance exam. Spermatogenesis is a multi-step process of sperm production. Germ cells progress from spermatogonia to spermatozoa through mitosis, two meiotic divisions, and morphological maturation. Why this matters? Fundamental for understanding fertility, endocrine regulation, reproductive pathologies across medicine, dentistry, and pharmacy. A common mistake is that confusing meiosis I versus meiosis II or underestimating Sertoli cell functions. Spermatogenesis is an intricately timed, hormone-dependent process taking place in the seminiferous tubules of the testes. Spanning roughly 64 to 74 days, it converts diploid spermatogonia into haploid, motile spermatozoa through mitotic expansion, two meiotic divisions, and differentiation, spermiogenesis. Mastery of this topic equips students to interpret male fertility assessments and understand hormonal therapies, recognize reproductive disorders. After studying this summary, you will be able to identify each cellular stage and its chromosomal content, explain endocrine control via GnRH, LH, and FSH, solve numerical problems related to cell output, and link disruptions in spermatogenesis to clinical infertility. Now, some key definition and concepts. Spermatogonium is a diploid stem cell on the basal lamina, divides mitotically to self-renew and create primary spermatocyte. A primary spermatocyte is the first meiotic cell, 2 and 4 cDNA, initiating prophase 1. Secondary spermatocyte is a product of meiosis 1N, 2 cDNA, quickly enters meiosis 2. Spermatid is a haploid 1N, 1 cDNA cell that begins morphological maturation. The spermatozoon is a fully mature sperm cell with head, midpiece, and flagellum. Sertoli cells are the supportive nerve cells forming the blood-testis barrier, secreting inhibin, beta, and responding to FSH. Leydig cell is an interstitial cell producing testosterone in response to LH. The blood-testis barrier is a tight junction complex protecting germ cells from autoimmunity. Spermiogenesis is a transformation of spermatids into spermatozoa, acrosome formation, and flagellum development. Spermiation is the release of mature sperm into the lumen of the seminiferous tubules. Checkpoint question. Describe how the chromosomal content, N/C, changes from spermatogonium to spermatozoon. Spermatogonial mitosis. Type A dark spermatogonial cell renew. Type A pale become type B, which differentiate into primary spermatocyte. Meiosis I, primary spermatocyte to secondary spermatocyte. Primary spermatocyte 2 and 4C undergo prophase 1, metaphase 1, anaphase 1, telophase 1. Form two secondary spermatocyte, 1 and 2C. Meiosis II, secondary spermatocyte to spermatids. Secondary spermatocytes rapidly complete meiosis II, producing 4 spermatids, 1 and 1C. Spermiogenesis is a morphological maturation. Spermatids elongate, condense their nucleus, form acrosome, assemble microtubule-based flagella, and package mitochondria in the midpiece. Spermiation is the release. Spermatozoa detach from Sertoli cells and move into the epididymis for final motility acquisition. Example of calculation. Given 2 times 10 to the power of 6 primary spermatocytes begin meiosis 1. Formula. 1 primary spermatocyte is 4 spermatozoa, as we said. So you just have to multiply this number by 4, which is 8 times 10 to the power of 6, which is 8 million spermatozoa. Common misconception is that meiosis 2 further halves chromosomes number. Actually, ploidy reduction occurs in meiosis 1. Meiosis 2 separates chromatids. Some examples. 500 spermatogonia enter differentiation pathway. How many secondary spermatocytes form? We know that the spermatogonia become primary spermatocytes, 1-to-1 ratio. However, the primary spermatocytes to become secondary, they differentiate into 2. So 500 primary spermatocytes times 2 would lead to 1,000 secondary spermatocytes. Exogenous testosterone therapy lowers GnRH. Predict the effect on spermatogenesis. Low GnRH, low luteinizing hormone, low intratesticular testosterone, thus impaired Sertoli support, oligospermia, which means reduction or decline in the production of sperms. You must remember these for the exam. One germ stem cell yields 4 haploid sperm cells in almost 70 days. Sertoli cells under FSH form the blood testis barrier and secrete inhibin beta. LH stimulates Leydig cells. Testosterone concentration in seminiferous tubules must remain high for meiosis completion. Clinical conditions such as varicocele, cryptorchidism, or chemotherapy disrupt spermatogenesis. In the entrance exam, also remember to check the test cell counts, hormonal axes, chromosomal content at various stages. These are important because they're always asked upon. Some mnemonics to help you remember. Someday some sperm survive, where we go from spermatogonia to primary spermatocyte to secondary spermatocyte, spermatid, and spermatozoon.